Assalamu alaikum, dear students. I am Dr. Nadia Taimur, Assistant Professor, Department of Botanical and Environmental Sciences, CUST. So today we will discuss the topic which is development and types of ovules from our course Introduction to Embryology of Angiosperms. The outline of today's lecture includes three main topics: development of ovule, structure of ovule, and types of ovule. So the question arises, what is an ovule? In seed plants, the ovary contains one or more roundish or oval egg-like bodies called ovules. Each ovule contains a large oval cell called the embryo sac. The ovary give rise to fruit and the ovules to seeds. This image represents typical structure of an ovule, and this is the embryo sac which is present inside the ovule. Now let's discuss how does an ovule develop. The ovule arises as a small mound of homogeneous tissue on the inner wall of the ovary. It grows rapidly and evolves into a prominent conical structure with a rounded tip. This mound develops to form the central part of the ovule called the nucellus. This image shows various stages of the development of an ovule. So as the development continues, either one or two layers of tissue evolve from the base of the nucellus, forming the integuments. As you can see here, these are the two integuments that are in the process of development. So the inner integument develops first, and after that, the second one evolves. The growth of integument is much faster than the nucellus. As you can see here, these integuments are growing faster than the nucellus. The integuments completely enclose the nucellus except for a narrow opening called the micropyle, which you can see here and here and here. So now let's discuss structure of a typical ovule. This is the diagram of a typical ovule. Each ovule is attached to the inner wall of the ovary by a slender stalk which is shown here called the funiculus. Now the point of attachment of the ovule to the funiculus is known as hilum. So this portion represents the hilum. The main body of the ovule is formed by the central mass of parenchymatous tissue, and this central mass of parenchymatous tissue is called the nucellus. The nucellus serves to cover and provide nutrition to this embryo sac. Now, each ovule has two distinct ends, the micropyle end and the chalaza end. Externally, the nucellus, which is shown here, is covered by one or two protective covers called the integuments. So these two here represents the integuments. Now there are different types of ovules depending upon the number of integuments. When only one integument is present, such ovule is called unitegmic ovule. In some cases, two integuments are present. Such ovules are known as bitegmic ovule. Very rarely, three integuments are present, and such ovules are known as tritegmic ovules. In some cases, no integument covers the nucellus, and in such cases, the ovule is known as ategmic ovule. Now, in an ovule, the most important structure is the embryo sac, which is shown here. Female gametophyte is another term used for the embryo sac. The embryo sac consists of the following structures, including egg cell or female gamete, synergid cells, antipodal cells shown here, and polar nuclei. Now let's see the structure of an embryo sac. So this image represents different cells present inside the embryo sac and their functions. As discussed in the previous slide, the embryo sac contains antipodal cells, polar nuclei, egg apparatus, which contains the egg cell and the synergids. Antipodal cells are present towards the callosal end of the ovule. The antipodal cells, they either remain unchanged or they degenerate or they proliferate in number or they develop transverse cell walls as in maize, and sometimes they become hostorial. The polar nuclei present in the middle of the embryo sac, they either fuse or remain separate but paired. The egg apparatus is present towards the micropylar end, and it also has structures called filiform apparatus. This filiform apparatus is very important because it absorbs nutrients from the walls, secrete chemotropic substances to attract pollen tube, and is the target for the pollen tube entry. Now we come to our next topic, which is types of ovules. On the basis of position of micropyle with respect to the chalaza and funiculus, ovules are of the following types. These include Orthotropous ovules, Anatropous ovules, Hemianatropous ovules, Campylotropous ovules, Amphitropous ovules, and Circinotropous ovules. So let's discuss first Orthotropous or Atropous or straight ovule. Orthotropous ovule is the most primitive and simplest of ovules. The ovule is erect or straight so that the funiculus, chalaza, and micropyle and hilum, they'll all lie in one straight line. Therefore, it is also known as straight ovule. Examples include members of family Polygonaceae and Peperaceae. The second ovule that we are going to discuss is known as Hemitropous or Hemianatropous ovule. This type of ovule is also known as horizontal ovule. In this type of ovule, the body of the ovule lie at right angle to the funiculus, as you can see here. So this is the ovule, and this is the body of the ovule, which is placed at right angle to the funiculus, so that the micropyle and chalaza, they lie in one line. Examples include members of family Ranunculaceae. So the next ovule is Anatropous or inverted ovule. Anatropous or inverted ovule is the most common type of ovule found in angiosperm families. Nearly 80% of the angiosperm families contain the Anatropous ovule. Now, in this type of ovule, the whole body of the ovule, as you can see, is inverted through 180 degrees. And due to this, the micropyle lies close to the hilum here. So, Anatropous or inverted ovule is the most common type of ovule and is present in more than 80% of angiosperm families. In this type, the ovule bends along the funiculus so that the micropyle lies close to the hilum. So the whole body of the ovule is inverted through 180 degree. As a result, the micropyle comes close to the funiculus. This type of ovule is commonly found in both monocot and dicot families, and notable examples include Asteraceae and Solanaceae. So the next type of ovule that we are going to discuss is the Compylotropous, or it is also known as curved ovule. So the whole body of the ovule is bent or curved. Now, due to curvature in the ovule body, the micropyle and the chalaza, they do not lie in the same line. So the alignment between chalaza and micropyle is lost. The embryo sac is only slightly curved, not a lot. Notable examples include members of Brassicaceae and in particular Capsella. So the next type of ovule is called Amphitropous or Transverse ovule. In this type of ovule, the body of the ovule and embryo sac is bent or curved. So in this case, both the embryo sac and the ovule, the body of the ovule is curved. Now, because of this curvature, the embryo sac is bent like a horseshoe. So the horseshoe shaped embryo sac is the characteristic feature of this type of ovule. Now, this type of ovule is commonly found in family Laminaceae, and other notable examples include Poppy, Alisma. Now, the last ovule, which is known as Circinotropous ovule. So, in this type of ovule, initially the ovule is orthotropous or straight. But as the development continues, the ovule becomes Anatropous due to unilateral growth of the funiculus. This is the funiculus. This growth continues until the ovule once again becomes straight or orthotropous. As a result, the funiculus completely surrounds the body of the ovule. Due to this coiled structure, the ovule is also known as coiled ovule. So during development, the ovule was initially straight, then become inverted, and then became straight again. So the most important feature of this ovule is that the funiculus surrounds the whole body of the ovule, and for this reason this ovule is also known as coiled ovule. This ovule turns at 360 degree during development. Notable examples include members of family Cactaceae. So we have discussed different types of ovules depending upon the position of micropyle in relation to chalaza and funiculus. And we discussed 6 different types, which included Orthotropous ovules, Hemitropous ovules, Anatropous ovules, Compylotropous ovules, Amphitropous ovules, and Circinotropous ovules. This last slide of the lecture contains references to sources from which the material is taken and made into lecture. I hope it was helpful. Thank you.